Cytoplasm microtubules threads cytoskeleton. For illustration, location: it fills the space between the cell membrane and the nucleus. Structure: it is a fluid-like, semi-liquid substance that is mainly composed of water. Some organic and inorganic substances. Contents: it contains cytoskeleton. It is a network of threads and microtubules, which provides the cell with a support that helps in maintaining its shape and form. Works as passages that transport different substances from a place to another inside the cell. Cell organelles. They are a group of various structures which are divided into
Ribosomes. Description: they are non-membranous round-shaped organelles. Location: some of them are free in the cytoplasm, single or in clusters, the least number. 2 others are attached to the outer surface of the endoplasmic reticulum, the largest number. To produce and release protein directly into the cytoplasm, so that the cell uses it in its vital processes, such as growth, regeneration and others. To produce proteins as enzymes that are transported by the endoplasmic reticulum to outside the cell after making some modifications to them in the Golgi body. Function: they are responsible for synthesizing protein in the cell.
Centrosome. It is located in animal cells, except the nerve cells, and some fungal cells near the nucleus. It is not present in the cells of the plants, algae and most fungi, whereas these cells contain a region of cytoplasm instead of the centrosome which performs the same function. It consists of 2 tiny particles called centrioles or centrosome. Each centriole consists of 9 groups of microtubules that are arranged in triplets in a cylindrical shape. It plays an important role during the cell division, where the spindle filaments extend between the 2 centrioles that are present at each pole of the cell. So, these centrioles withdraw the chromosomes towards the cell poles in order to help in the cell division into 2 cells. 2. In the formation of flagella and cilia, movement tools in some unicellular organisms. Key points: ribosome and centrosome are non-membranous organelles, therefore they are the least affected by lipid solvents. Spindle filaments are proteins that have the ability to shrink, allowing the chromatids attraction towards the cell poles. Centromere. The point of connection of the 2 chromatids. Centrosome. It consists of the 2 centrioles. Centriole. It contains 27 microtubules as shown in the figure, 9 groups of microtubules x3. During the cellular division, the centrosome duplicates to give 4 centrioles. Each cent directs to one of the cell poles to make the spindle filaments extend from it.
Membranous organelle 1. Endoplasmic reticulum. Description: it is a network of membranous canaliculi. T extends through all the parts of the cytoplasm and attaches to the nuclear envelope and cell membrane. Functions: it forms an internal transport system that benefits in transferring substances from a part to another inside the cell. It transfers substances between the nucleus and cytoplasm. Types: there are 2 types of endoplasmic reticulum, which are POC. The presence of ribosomes. Functions: sites of abundance. Tout rough endoplasmic reticulum. It is characterized by the presence of a large number of ribosomes on its surfaces. 1. Synthesizing proteins in the cell. 2. Making modifications on proteins that are produced, secreted by ribosomes. 3. Making new membranes in the cell. The stomach lining cells, as these cells are responsible for the secretion of digestive enzymes. The cells of endocrine glands, as these cells are responsible for the secretion of hormones, proteins. Smooth endoplasmic reticulum. Ribosomes are absent from it. 1. Synthesizing lipids in the cell. 2. Transforming glucose into glyco.
Golgi body, Golgi apparatus. Description: it is a group of flat membranous round-ended sacs. Number. The number of Golgi bodies in the cell differs according to the cell's secretion activity, where it increases in the glandular cell. Function: it plays an important role in the formation of the cell secretions, where it performs its function in several stages, as follows. It receives the molecules that are secreted by the endoplasmic reticulum through a group of transporting vesicles. 2. It classifies and modifies these molecules of substances. 3. It distributes these substances to the places where they are used inside the cell, or it may pack them inside secretory vesicles called lysosomes, that move towards the cell membrane, where the cell expels them outside as secretory products.
Lysosomes, secretory vesicles. Description: they are small, sized, round-shaped and membranous vesicles, where they are formed by Golgi bodies and contain inside them a group of digestive enzymes. Lysosomal enzymes. Functions: getting rid of worn out and senile cells and organelles which are useless to the cell. Digesting large molecules of nutrients that are engulfed by the cell and converting them into structurally simpler substances to enable the cell to make benefit from them. Note: the cell is not affected by the enzymes of lysosomes, because these enzymes are surrounded by a membrane that isolates them from the cell components. Example: white blood cells, corpuscles, use the digestive enzymes that are present inside the lysosomes to digest and destroy pathogens, as microbes, which invade the cell. As shown in the following figure.
Mitochondria. Description: they are sac-like membranous organelles. Their wall consists of two membranes, outer and inner. A group of folds known as cristae are extended from the inner membrane into its matrix. They are considered the main storehouse for the respiratory enzymes in the cell. They are the storehouse for the substances that are necessary to store the energy resulted from the cellular respiration, as a result of nutrients oxidation, especially glucose, where this energy is stored in the form of ATP compounds, adenosine triphosphate, and the cell can extract this energy from these ATP compounds once more. So, the mitochondria represent the centers of the energy production in the cell, the energy storehouse in the cell. Notes: 1. The function of cristae. They work on increasing the inner membrane surface area on which the chemical reactions that produce energy take place. Needed by the muscles. 2. The number of mitochondria increases in muscular cells to increase the production of energy.
Key points: ATP molecules are considered the energy currency inside the cell. because they represent intermediate molecules circulated among the reactions that produce energy such as cellular respiration and the energy-consuming reactions such as movement and protein synthesis. There is a direct relation between the number of cristae inside the mitochondria and the amount of produced ATP molecules. The more number of cristae inside the mitochondria, the more amount of energy produced. ATP number of crista 5 vacuoles description. They are membranous sacs that resemble bubbles filled with a fluid. In animal cells, they are small in size and large in number. In plant cells, they are gathered in one big vacuole or more called sap vacuole. Function: storing water and nutrients, or storing wastes of the cell, until it gets ring 2 plastids description. They are various shaped membranous organelles. Location: they are present in plant cells only. Types: there are 3 types of plastids that differ depending on the type of pigment that is present in each type of them, which are type of pigments, function, location. Leucoplasts, white or colorless plastids. They don't contain any type of pigments. They work as centers for storing starch. They are present in the root cells of sweet potatoes, tubers of potatoes and internal leaves of cabbage. S2 chromoplasts, colored plastids. They contain carotenoid pigments whose colors they contain the green are varied between red, chlorophyll pigment, yellow and orange. They give the plant or its parts a characteristic color. They are present in the roots of some plants such as rapeseed, also found extensively in the petals of flowers and fruits like tomatoes. 88 chloroplasts, green plastids. Photosynthesis occurs in them, where chlorophyll pigment transforms the light energy of the sun into chemical energy that is stored in the chemical bonds of glucose. They are present in the leaves and stems of green plants. Structure of the chloroplast matrix, stroma, outer membrane granum. Chloroplast, diagrammatic figure, Chapter 2. Matrix, stroma, chloroplast under transmission electron microscope. It is composed of a double envelope membrane, which consists of outer and inner membranes. An internal matrix called stroma, inner membrane compact layers of disc-shaped structures known as thylakoids, where each group of them forms what is known as granum, which is found inside the stroma. Notes: carotenoids, carotenes. They are colored pigments whose colors are varied between red, yellow and orange, where they are found in the chromoplasts of the plant cells. Chromatin. It is minute tangled filaments that are coiled around each other and found in the nucleus of animal and plant cells. Lesson T for estration. Only the colors of plant cell are related to the presence of chromoplasts as in the petals of flowers, or to the presence of some colored pigments in the cytoplasm as in beet and roselle.